This week, Lab TV travels to an Air Force research lab in San Antonio, Texas, where scientists are developing and testing tools that can find the creepiest bugs on the planet.
Some of the naturally occurring organisms are your Salmonella, your E. coli. You also have Anthrax, your plague. But we want to test for biological organisms because some of these organisms that are naturally occurring can make us sick, but some of them can also be used to harm or become a threat to us.
These organisms are called pathogens because they cause diseases. Sometimes they're found in food or in the ground, but the real danger would be if they were used on purpose to hurt people. So if people are getting sick or a threat is suspected, scientists need to quickly figure out the cause.
It could just be a lot of people coming down with influenza, but if it is something else, one of your other threats, your anthrax or plague, you wouldn't know that until you actually test for the organism.
Let's say they're looking for Salmonella, a bacteria that causes food poisoning. Like every living thing, Salmonella has a unique DNA fingerprint. These scientists can find tiny amounts of Salmonella DNA by using a powerful process called PCR, polymerase chain reaction.
In the PCR reaction, we have a primer. The primer corresponds to the DNA sequence that we know about, like the food poisoning causing organism. It sticks to that at the right temperature because we've melted the DNA first by boiling it away at 94 degrees C. The DNA splits, the primers bind when it gets cool, and then the polymerase latches onto that and extends and adds an entirely new strand of DNA. Now taking the original single strand of DNA and making it double-stranded, one new copy. Then the next cycle, the process repeats, the temperature rises, the two strands split apart, primers bind to both strands, cools down, reaction proceeds, and we get another set of DNA. Now we have 4 strands. This repeats. 4 strands begets 8 strands begets 16, and so forth.
PCR is like a copying machine. If the primer matches the organism's DNA, PCR will build millions of DNA copies. And the reason why it works is an enzyme called polymerase.
Well, polymerase is isolated originally from organisms that were adapted for living in thermally hot environments, such as deep sea vents or some of the hot springs in Yellowstone.
That organism, Thermus aquaticus, loves hot water. So its enzyme, polymerase, doesn't mind the near boiling temperature needed for the PCR process. The Air Force can use PCR to test for pathogens anywhere in the world with a very James Bond-looking device called the JBAIDS.
And the JBAIDS is ruggedized, which means we can take it, we can put it on an airplane, we can throw it out of an airplane, we can get it to our field personnel, and they can use this.
This is the JBAIDS, and it gives the Air Force the capabilities to detect pathogens in the field. Open it up, plug it in, and it's ready to go. You will load your sample. You'll take a small amount of it, load it onto your capillary, and once you have your carousel loaded, you can see these are glass capillaries. All the PCR reaction will take place in the capillaries. Close the lid and then press start. And within 45 minutes, you'll have an answer.
It used to take several days to identify a pathogen, but now with PCR, it can be done in less than 2 hours.
48 hours is pretty quick, but that's long enough for people to have been exposed to an organism and begin to see symptoms, maybe to leave an area. It'd be much better to catch it quickly in 2 hours and be able to call someone up on the phone and say, hey, you've got a problem.
Finding a problem fast could mean saving thousands of lives. And that's why these scientists keep testing and making these tools better every day. To find out more about molecular biology, pathogens, and PCR, check out labtvonline.org.